Welcome, everyone, and good morning. It is our pleasure to host and welcome you to the 15th annual meeting of the Indiana Clinical and Translational Science Institute. The theme of our meeting is Translational Neuroscience, Transforming Health Through Innovative Research, and that is indeed what you will hear all day today. So I'm Sharon Moe. I am the co-director of the CTSI with my colleague, Sarah Wehe. I'm also the Associate Dean for Clinical and Translational Research at the IU University School of Medicine and the Division Director of Nephrology and Hypertension.
Hi, and welcome to everyone. I'm Sarah Wehe. I'm the Associate Dean for Community and Translational Research at IU School of Medicine. And so pleased to be here with over 300 individuals who have RSVP'd to be here in person. I'm sure many will be filtering in here, and over 150 online, a record attendance. So we're all very excited for the day ahead of us.
Right, just a couple of housekeeping details. So please take a minute to mute your cell phone. I know it's always one of those things we got to think about. And just the women's restroom is out to the right, the men's is out to the left. This meeting is also eligible for continuing medical education, and you will notice in the slides in between a QRS code that you need to scan that will actually register you to get the email to actually then complete the evaluation to get the CME. Got that? So QRS code, register, or you could step up at the desk and tell them that you're very interested in that. If you are, all of the biographies of our speakers and all of the conflict of interest are on our online or in the speaker book. So that's hers, this one, there we go, thank you. Get to this QRL. This is the CME information, and I'm gonna stop right here and let anybody that wants to scan for a second to get your CME. I don't see a lot of cell phones sticking up, but just a few. Okay. All right, I'll hand that over now to my colleague, Dr. Wehe, who's going to introduce you to what the CTSI is all about. Sorry, it wasn't. There you go. Here is, yeah, go ahead, Steph. This is the agenda. I'm supposed to introduce the agenda too. This is the agenda for the morning. We're going to have a very wonderful speaker who has been on campus for a couple of days, really inspirational presentations. She is Huda Zogby. And then we will have a morning kind of roundtable of activities from around the state. And then this afternoon, we will have a translational, sorry, I'm just going backwards here. Could you go to the next slide, please? It'll be the best way to do it. And then we'll have this afternoon, we will have a wonderful discussion up here, and then we're going to have a wonderful closing presentation as well. So thank you.
All right, so as many of you know, the Indiana CTSI is a part of a national network and uniquely statewide. There's over 60 CTSAs across the country. To really focus on increasing the efficiency and speed of clinical and translational research. And the CTSA, our CTSI, the Indiana CTSI, is the only one in the state of Indiana and one of the very few that encompasses an entire state. So translation is the process of turning observations in the laboratory, clinic, and community into interventions that improve the health of individuals and the public, from diagnosis and therapeutics to medical procedures and behavior changes. And the CTSI stands for the Clinical and Translational Sciences Institute, which is supported 4-to-1 beyond the NIH dollars with contributions of our many, many partners across the state. We are a partnership of Indiana University, Purdue University, and the University of Notre Dame. Thank you all for coming, as well as partners that include the public, as indicated in that first map, a new partnership that we're launching with Ivy Tech, with our treasured health system partners, as well as philanthropy and industry, and our many community organizations in every county, all of the 92 counties. And we are thankful to our partners across the state that we have successfully received our fourth renewal of our CTSI of $38 million for over the next 7 years. I'm clearly failing at the slide advancement. So our mission is to bring Indiana's brightest minds to solve the state's most challenging health issues. And we envision in Indiana that's one of the nation's healthier states. And we hope to embark on this in the next 7 years so that by 2030, we envision an Indiana CTSI that will transform our statewide research ecosystem with deliberate attention to diversity and inclusion in our partnerships, workforce development, and research engagement. And we will accelerate innovation and rapidly implement evidence into practice in order to advance health equity. And with that, I'll pass it back to Sharon.
So it's now my pleasure to introduce to you Dr. Dean Hess, Dean J. Hess, who will begin our morning session. For those of you who are new to us, you may not know all of his accomplishments, but Dean Hess is the 10th Dean of the IU School of Medicine, joining us in 2013. So the IU School of Medicine is the largest medical school in the country with 9 campuses, over 360 students, and 11,000 faculty and staff throughout Indiana. He is also the Executive Vice President for University Clinical Affairs, the Walter J. Daly Professor, and the Director of the Indiana Statewide Medical Education System. He's a busy man. During his tenure, he has overseen a doubling in research funding from the National Institutes of Health, has led the school through a comprehensive curriculum reform, and strengthened the relationship with IU Health, the school's primary clinical partner, and one of the nation's academic premier institutions. He himself is also a translational physician scientist who has authored more than 100 scientific papers and has made a significant impact in patient care. It is a pleasure to have him here to welcome another translational scientist who has also significantly impacted patient care. Thank you, Dean Hess.
Well, thank you, Sharon, Sarah, and it's a great privilege to be here again this year. The Watanabe Prize is named in honor of the late Gus Watanabe, a giant in the field of translational research in both academia and industry, who impacted the health of people around the world in a number of roles, including serving as a leader at Indiana University School of Medicine, Eli Lilly and Company, as well as the Central Indiana Life Sciences Initiative, which later became the organization that we know to be BioCrossroads. Because of Gus's unparalleled dedication to scientific inquiry and the tireless advocacy for translational research, this award was established to recognize outstanding contributions in translational research in those who have shepherded scientific discoveries into new therapies. Now, unfortunately, this is the first year that Dr. Margaret, or Peg Watanabe, Gus's wife, was not able to join us. She's out of town, but I'm sure she's here in spirit with us today. So at this time, it's a great pleasure to introduce Dr. Huda Zogby, a distinguished service professor at Baylor College of Medicine, the director of the Jan and Dan Duncan Neurological Research Institute at Texas Children's Hospital, and an investigator with the Howard Hughes Medical Institute. Dr. Zogby told me to keep the introduction really short because she has a lot to talk about, but suffice it to say, she's extremely well qualified for the Watanabe Award, as her research has impacted the patients in a wide variety of areas, rare and debilitating diseases, including Rett syndrome and many other neurologic diseases. So I'll cut it short and you can come on up. And I'd also like to welcome Dr. Tatiana Faroud to the stage. Dr. Faroud is our Executive Associate Dean for Research Affairs at the IU School of Medicine, a distinguished professor, chancellor's professor, and importantly, the August M. Watanabe Professor for Medical Research. Dr. Faroud's a statistical geneticist and a leader in dementia research who leads a large NIH-designated repository for samples from patients with Alzheimer's disease. And as our EAD for research affairs, Dr. Faroud's also responsible for the administration of this prestigious award. So, Dr. Zogby, I'm honored on behalf of the entire research community here to present you with the 2023 Watanabe Prize and plaque honoring you as the 2023 awardee. I think we'll probably move out here. This is important, the check.
And everyone, please take a minute and come down and fill the seats.
Good morning, everyone. It's truly a huge honor to be receiving the Watanabe Prize, receiving an award in an honor of a human being who is passionate about bringing the best science to patients and about mentoring others and inspiring them to do scholarly work to really advance health and therapeutic is very inspiring to me and will be a reminder of what we do, why we do what we do every day. It's also a huge honor to really receive an award from Indiana University after I spent a day here meeting various faculty, trainees, and members of the leadership team. It just showed me what a special place this medical school is and the research infrastructure that you've built to really reach health advances to all in the state. Thank you so much. I will hold this very dear and we'll keep up hopefully with all that's going on. And my first thing I'm going to do after this visit is send one of my best trainee. I have one already here you'll hear about, but send one of my best trainee to you hopefully in the near future. Thank you. So I was going to jump into the science, but Dr. Faroud asked me to take 5 minutes just to share my research path with you. And it wasn't prescribed. It was really mostly shaped by serendipity and just passion and observations. I started as a pediatric neurologist. I wanted to be a clinical pediatric neurologist, but in my first year of training, I encountered a patient with Rett syndrome, a neurodevelopmental disorder where the girls are born healthy and gradually lose all the milestones. And essentially every part of the brain is affected. All symptoms you've seen in any neurologic disease are combined in this disorder. And this is where serendipity happened. There were nobody knew about this syndrome in the US. It was reported in the Annals of Neurology the same month. I saw my first patient where 35 girls from Europe were described. So by chance, I saw the first girl in the US, and a week later, I saw another girl with a diagnosis of cerebral palsy, wringing her hands, having the same disorder, and then was able to find a few more by reviewing medical records. And that's where I was inspired to do research. I was still early in my training. I finished the neurology training and wanted to really figure out what causes this disease. And that's what got me into science. It's as simple as that. I was fortunate that Arthur Baudet took me on. We're the product of our mentors, our collaborators, and our trainees. And you'll hear in my story that's amplified many times. I have to admit, having come from Lebanon, and I'm backing up now, having come from Lebanon during the Civil War, applying late to medical schools in October when all schools started, all schools rejected me, but Meharry Medical College and a dean there took time to listen to my story and accepted me. And that's why I ended up graduating from Meharry Medical School. I had challenges being accepted in residencies, but again, Ralph Fagan took me into pediatrics. So that's really what started my career and the opportunities. And then Art, my scientific mentor, gave me the opportunity, someone who doesn't even know what's an exon and what's an intron entering his lab. He gave me a chance to learn basic science in his lab. However, in 1985, right before PCR was discovered, we had nothing about the genome, we had very little tools, and Rett is a sporadic disorder. So you couldn't really map the gene using family. It's one in a family. And Art looked at me and said, no way you can work on Rett because you're not going to get anywhere. So he insisted I pick a Mendelian disorder. And he was generous. His lab was working on urea cycle defect. He's a giant in that field. But he let me bring an adult neurologic disorder to his lab and worked on spinocerebellar ataxia type 1. And finished my postdoc in 3 years and started my own lab thereafter. And while I continued to work on Rett syndrome, I'm thankful to Art for advising me because, as you'll see from our studies, the SC1 project was moving smoothly. It took 16 years to find the Rett syndrome gene. That's what happens when you don't have the genome. And now what took us 16 years can be done for the trainees in the audience in 5 to 6 hours. So there's plenty time to do research because the discovery will happen much faster. I will just say one thing. When I started my lab, I really focused on being in the lab. I got a lab without an office, which I think was a blessing because that means I was working at the bench with my trainees, and that really enhanced my productivity. I also think having 2 children under 3 years of age when I started my lab was extremely helpful because that meant I didn't travel. I just went to 2 national meetings a year and I really focused on the work. And that, I would think, those 2 factors, plus the factor of growing up in a generous, collaborative environment, is really what shaped my career. And that allowed me to mentor trainees and the lab to grow with some of the best trainees, which, as you'll see from my talk, they're really now what drives me to come to work every day, and they are the future of research. So with that, I will delve into the science, and you'll see some of the aspects I shared with you as we discuss that. So spinocerebellar ataxia is what Art allowed me to work on in his lab because it's a Mendelian disorder, so you could map a gene. It's a dominantly inherited disorder, extremely rare, 1 in 100,000, characterized by loss of balance and coordination. Only if you have younger onset, you'll see cognitive deficits, and eventually the patients who will be healthy till they're about 40 years of age, they'll gradually deteriorate till they are wheelchair bound, then they'll have swallowing difficulty, and sadly that's what causes choking, and pneumonia what causes premature death. So we mapped the gene and discovered that in Minnesota, Harry Orr was working on another family with a similar disorder. So I reached out to him that we collaborate rather than compete, which was uncommon back when discovering a disease gene was hot. But I think that was one of my best scientific decisions, and Harry would agree for himself as well, that we joined forces and worked together. And we discovered the gene on the same day in our respective laboratory. And the mutation is an expansion of 3 bases of DNA within a protein we named ataxin-1 because we knew nothing about the protein, and it was the very first ataxia gene discovered. And now there are over 190 different ataxia genes. And just to highlight, the disease affects predominantly the cerebellum, as you see the atrophy here, and you can see the atrophy of the brainstem. So it's predominantly brainstem and cerebellar degeneration, but there's some loss in the hippocampus. So what I'm going to share with you today in my talk is the journey from finding a disease gene to really understanding the mechanism of disease. And understanding the mechanism of disease is really at the core of translation. I always use the analogy of language. If you want to translate a piece of literature at a high level, from say English to French, Arabic to English, you have to be a master of both languages. So for us to translate and bring good therapies to patients, we have to master understanding of the basic mechanism of disease. You have to understand something very well in order to translate it. So I'll share with you that. And through that discussion, I will highlight some things we learned from studying this really rare disease that informed us about how we're going to have to go about therapy. And it also informed me, at least inspired me, to move on and study the more common adult neurodegenerative diseases such as Alzheimer's. So the first thing you see when you have a mutation, you want to know, is it killing the function of the protein or is it affecting it in a different way? So to do that, you create a loss of function, which means you delete the gene in the mouse and you see, do you reproduce the features of the disease? And the answer was no. When we removed this gene from the mouse was fine. The only thing this mouse had is some learning and memory problems, but it did not have any ataxia or Purkinje cell degeneration. So that clued us in then that maybe it's not loss of function. It could be gain of function. It could be this protein with this mutation is gaining some function. And for this, we created a mouse where we put in the expanded glutamine repeat in the mouse locus. So now we're expressing the mutation in the right time at the right place, rather than artificially adding extra copies of the gene and so on and so forth. So we had a great model, and this model reproduced all features of spinocerebellar ataxia type 1. So now that you have a good model, you can answer many questions. What causes regional vulnerability? What's the mechanism of the disease? While we were doing this, in parallel, Harry's lab was studying the effect of the mutation just on Purkinje cells. And for that, he did use an overexpression model. And you could see in this case, when he put the 82 expanded repeat, you don't have to have as long repeat when you're overexpressing a mutation. You'll see loss of Purkinje cells. But one important discovery he made is that he found out that if you change one amino acid in the protein ataxin 1 that normally is phosphorylated, that now cannot be phosphorylated, even though you still have the human mutation, the protein is no longer toxic. And that was really very important discovery. And as you can see, you'll see through the talk, Harry and I go back and forth. Having seen this discovery from his lab, we followed up on it to try to understand, what does that phosphorylation site do? And what we discovered, this phosphorylation site leads to binding to another protein. And this particular protein, when it binds ataxin 1, it halts this degradation. So now we know why when he mutated that serine to an alanine, no longer binding, the protein can be degraded. We also learned that the glutamine expansion enhances the binding to this protein. So now the picture is emerging where what's happening is when you have a long glutamine, you enhance binding to 4-CT3, you stabilize ataxin 1. So that's a hint that maybe the gain of function is due to a little bit too much ataxin 1. And if that's the case, if we reduce this protein, maybe we can help. And the answer was yes. You'll see here, if you take one copy using a mouse lacking one copy, we got from Anthony Winshaw-Boris, you decrease ataxin 1, both the wild type and the mutant, by about 20%. You'll see the reduction here and here, here. And that is sufficient to rescue Purkinje cell degeneration. So you could see here's a healthy mouse, here's the knock-in SCO1 mice, you see loss of dendrite, loss of some Purkinje cells, and here when we just reduce the protein by 20%, we now have a rescue. So having discovered it's simply about the normal function of the protein and its accumulation, we wanting to know, well, what's the normal function of this protein? What does it normally do? And to that, we tried first focus on finding who interacts with this protein, because I told you it's a protein with unknown function. And with that, we discovered it interacts with a particular protein called Capicua that is known to bind DNA. It was discovered in fruit flies, but then by Casanova lab. But then we also learned about it, that it also exists in mammals. We discovered it interacts with ataxin-1. We also discovered that ataxin-1 has a paralog that's a very similar protein in the human genome. We call it ataxin-1-like. The two are similar. They share this domain. It's called ataxin-1 homology domain. Not helpful, but it tells you it's a domain in both proteins. And only ataxin-1 has the glutamine tract. And Capicua, which bind DNA to repress gene expression, has two isoforms, and both of which interact with ataxin-1. We discovered those two bind together in the brain. They're the most preferred interactors in brain tissue, highly abundant in the cerebellum, but they're widely expressed. So with that, we wanted to revisit the idea, could there be any loss of function contributing to the phenotype? Because now we have a paralog. Maybe if you only knock out ataxin-1, you don't get a problem, but maybe you have to knock out Capicua or knock out both paralogs. And we did that, and I'm just going to summarize here to show you. If you knock out both ataxin-1 and 1-like or Capicua just in the cerebellum, because if we knock them out everywhere, they both are lethal. So we know that these are essential protein for development. But you see normal survival, normal activity. This is a rotating rod where we put the animals to measure their coordination and follow them. And you see totally normal coordination. And even at 12 months, absolutely no change. This is work done by Max Russo, a postdoc in the lab, and Tyler Chamberlain, a graduate student in Harry's lab. We pooled efforts to do this. And more importantly, absolutely no Purkinje cell degeneration. So we clearly don't have any contribution of loss of function of this protein in the cerebellum contributing to disease. And now we can focus on perhaps this gain of function. So I told you these two interact to repress transcription. So you go and look at gene expression in the cerebellum. And lo and behold, what we found is that in disease, the Capicua target, the genes that Capicua repressed, are over-repressed. So what is the gain of function? You're accumulating ataxin-1, you're having too much of the ataxin-1 Capicua function, so you're getting over-repression. All of these studies then, what we learn is that this complex, while it has important role in development, it doesn't really contribute, its loss does not contribute to SC1. I don't have time to show you the data, but we discovered that loss of one allele of Capicua can cause a new developmental disorder, autism, intellectual disabilities, and so on, and seizures. We call it Capicua haploinsufficiency syndrome. So that was interesting, and we discovered that first in mice, and that led us to the human disease. And now we know that the poly-Q expansion stabilizes the protein, leading to neurodegeneration probably. So with that, then the potential translational strategy is lowering ataxin-1 or disrupting the interaction. And I'm going to share with you stories about both. To lower ataxin-1, we identified genes that can regulate ataxin-1 level and wanted to see if any of them a druggable target. And we wanted to find ways to disrupt the Capicua ataxin-1 interaction. To find regulators of ataxin-1, we've done genetic screens, first in cells using fluorescent assays, and in parallel, in collaboration with the BOTAS lab, in fruit flies, where the fruit flies, if you express mutant ataxin-1, the eye degenerates. And then we take what's shared with both and test it in human neurons and in mice. So Jihye Park and Ismail Ramahi started the screens, and then Won-Sok Lee did a broader screen, genome-wide, and this led us to many candidates. And we've pursued some of these candidates. I would share with you a story that was quite revealing and humbling. We identified two kinases that if we inhibit either, we lower ataxin-1 level. It turns out they phosphorylate the serine. So many years later, almost 10 years later, we come back and discover that serine, they're kinases that regulate it, and if you inhibit them, you're going to lower ataxin-1. So that is an ideal therapeutic target, right? However, unfortunately, we discovered MSK1 only helps the cerebellar phenotype, and RSK3 mostly help the brainstem phenotype. So that creates a complication. And it turns out there is a reason for that. The reason being RSK3 is predominantly expressed in the brainstem in healthy mice and the knock-in mice. You see very little of it in the cerebellum, whereas MSK1 is predominantly in the cerebellum and very little of it in the brainstem. So no wonder when we knock out the genes in one or the other, we only get regional. And this is important because if we didn't do this kind of study, we would then foolishly run after one kinase inhibitor and we'll be met with failure, and somebody will conclude mouse studies are not helpful, not translatable to human studies. Mouse studies are really helpful. It's just that the humans need to do thorough studies. So with this, we then discovered two more additional kinases. And that's when we decided to give it up on the kinases. There are going to be 4 kinases phosphorylating that one serine in different tissues, not tractable. But we were interested, how good is that serine? Is it really going to answer all the phenotypes of the disease? Because Harry, as if you recall, only did the experiment in Purkinje cells, and the disease affects many cells. So Larissa Nietzsche in the lab, wonderful graduate student, created a knock-in mouse. Now we put the mutation on the knock-in expanded allele. So now we can see the effect of this mutation throughout the brain. And consistent with all earlier data, replication is good. We reduced ataxin-1 in all brain tissue. So this was the moment of truth. Are we going to rescue all phenotypes? And the answer was, we rescued the cerebellar phenotype. Not as beautiful as isolated Purkinje cells, but good enough. You can see here the performance improved. Slight improvement in survival, but unfortunately, there was no benefit in the hippocampal phenotype. So this told us the mechanism is not all through the serine, through every tissues. We also wanted to see if you had to have a small molecule that decreases this phosphorylation, it's not going to selectively in the brain work on the protein from the mutant allele, it'll work on both proteins. So we wanted to see what will happen if we have the alanine on both alleles. So we now mutated it on the wild type allele. So now both ataxin-1s cannot be phosphorylated there. So you're lowering both a little bit. And we asked what happened. And this is where a surprise came. And I think this has actually important translational consequences. What we discovered, the benefit we saw from selectively lowering the mutant protein is lost when you lower both of them, both in the rotarod, both in the survival. This is really important because when you're using a drug, whether it is to inhibit this phosphorylation or to just lower ataxin-1 just a little bit, it taught us that the wild type protein, while its knockout is totally inconsequential in the cerebellum, but partial lowering in the context of another mutant allele is consequential. So context matters. And this told us that the wild type protein is probably competing the mutant protein from its interaction. And that's why it's subduing the disease. And now just partially lowering both is not good enough. More importantly, James Orengo made a special knock-in allele that's conditional. And in the process, by inserting LOXP site, he inadvertently lowered mutant ataxin-1 by 50%. So here's only lowering the mutant allele. And this knock-in is our original. All the animals die by 52 weeks. And now if we simply lower just the mutant protein, the animals will live beyond 80 weeks, almost that. So it showed us that really, if we're going to do therapeutics for this disease, we have to either lower the mutant allele selectively, or if we're going to lower both, perhaps we have to do a lot more in a specific tissue. But we have to be careful, as I'm going to show you in additional studies we learn. So now we wanted to see, well, how about disrupting ataxin-1 Capicua interactions? And we mapped those interactions, and we collaborated with Jijun Song at KAIST, where we got it down to the amino acids on ataxin-1, 2 amino acid, and 1 on Capicua. And Harry now went back to the Purkinje cell studies, mutated the interaction domain. So now ataxin-1 cannot interact with Capicua and Purkinje cells. So here's the mutant ataxin-1. You see massive decrease in performance. But when you prevent interaction with Capicua, beautiful rescue, the same with the pathology. So really, preventing interaction with Capicua and Purkinje cells rescued that. So everything, working exactly as we would have suspected from our model. But then this is, again, just Purkinje cells. So a graduate student, Stephanie Coffin in my lab, decided now to put this mutation on the knock-in allele to see would this happen in all tissues. And you'll see here, if you IP Capicua, you can have, if you IP Capicua, you'll bring that mutant ataxin-1 and wild type. But if we mutate the interaction domain, we lose that. So now, if we disrupt those interactions, do we rescue everything? And the answer was, we do rescue some in the cerebellum, as you can see here. We don't rescue any learning and memory. And we have a modest, but not that impressive rescue of survival. So this immediately told us there is much more complexity to what drives disease mechanism in different tissues. When we looked in the Purkinje cell, the rescue was really, we did physiology with Roy Sillito. The rescue was really nice. The pathology was really nicely rescued. So this told us definitely Capicua is rescuing in the Purkinje cells, but in the rest of the cerebellum, there are other probably factors playing. And we studied the binding of Capicua on DNA. Remember, it's a transcription factor. It binds DNA. And Mark Durham helped with these experiments. And you'll see in SCA1 mice, knock-in mice, you see the increased binding. The wild type is black. And you'll see here, when you mutate the interaction, it gets back to normal. You'll see that here. But that getting back to normal only helped Purkinje cells. And when we look at the chromatin, for example, this mark that affects chromatin, that's not corrected. So that immediately clued that in disease pathogenesis is far more complex than we thought. We thought we found the interactor, we showed it in Purkinje cells, it's going to work everywhere, just disrupt that, you got the answer. Unfortunately, it's humbling. That's not the case. So we knew from work from Harry's lab that if you took ataxin-1 out of the nucleus, you got a beautiful rescue. So we knew the protein has to be a nuclear protein. And we then did study to identify interactors and found 3 putative transcription factors that we discovered their motifs are enriched among all the genes altered in our cerebellar SCA1 model. And now I'll just show you, this is candidates. We have to still now do more work to prove that. But only 10% of the cerebellar gene expression changes are targets of Capicua. And you'll see here that one of them, putative target of RFX, some are putative target of ZK-SCAN. We don't know the motif for ZBTB. Some are multiple transcription factor targets, and we still have some unaccounted for. And when we look at our rescue, when we eliminate interaction with Capicua, you'll see the hyper-repressed Capicua gene are corrected in blue here, but not the altered gene that are putative targets of these other. So this told us the disease is far more complex than we thought, and it may be different factors in different cell types and different tissues, and this is a direction the lab is pursuing now in a variety of tissues. So getting back to surprises in the disease and the importance of Capicua, Rudy Tanzi, a wonderful geneticist in Alzheimer's disease, reached out to us mentioning that he found SNPs in ataxin-1 that increase susceptibility to Alzheimer's disease. He also found copy number variation that span ataxin-1 in some patients, a handful of patients with Alzheimer's disease. So he and Ji Hong Song, a young assistant professor in his team, were curious to study the ataxin-1 knockout. Remember, those are mice we have sort of ignored because the disease is gain of function, But remember, I told you they have learning and memory deficit. That's the only thing. So we sent our mice immediately to Rudy, and Rudy immediately called me back and said, BASE1 is increased in the ataxin-1 knockout mice. And you can see on this plot, this is normal knockout heads. And also the A-beta cleaved product is increased, whereas the secreted alpha form is decreased. So when he called me about this result, I said, that's really not possible. And he said, it really is happening. And I said, I don't think so. I've been studying ataxin-1 for 20 years. I have tons of RNA-seq data at multiple stages of the disease. I've never seen BASE1 increased. Now remember, everything I showed you we do is in the cerebellum and hindbrain. We never really look at the cortex and the hippocampus as far as RNA-seq goes. So when we look at the cerebellum and brainstem, BASE1 is normal. So I said, I want to confirm this in my own hands. We did, and lo and behold, he was right. In the cortex, BASE1 is up. In the cerebellum, it's not. And if you look at this image here, which is immunohistochemistry for BASE1, knockout negative, this is wild type, and I hope you can clearly see that the protein is only increased in the forebrain. And if we were to look here, you'll see the same thing, that the BASE1 is really increased in these structures, but not in the cerebellum and not in the brainstem, and these are quantification. So this led us to collaborate together, and Larissa Nietzsche worked with them to figure out the mechanism. How is it happening? How is loss of ataxin-1 elevating BASE1? And I'm skipping through all of data. He showed that this aggravates the pathology from APP in the APP transgenic mice, increasing A-beta, and really causing more degeneration. And I'll summarize it here. Remember the two preferred partners, ataxin-1 Capicua? We knew from many studies that they repress this transcription factor, ATV. And what we discovered is that BASE1 is regulated by this transcription factor. And what we also knew from many studies in our lab, when you knock out ataxin-1, Capicua become unstable and it's decreased. So you have less Capicua, less repression, more ETV, upregulating BASE1. That's the mechanism. So this was the most humbling experience I had. Forget tissue-specific regionality being complicated. Now we're dealing with a protein whose gain of function in one brain region causes neurodegeneration and whose loss of function in another brain region make you susceptible to another neurodegenerative disease, Alzheimer's disease, which was really, really surprising and totally unexpected. And this is why I say it's a balancing act to treat to these diseases. So if we want to lower ataxin-1 using ASO or microRNA or combination strategy, we have to really make note that wild-type ataxin-1 is protective in some brain regions, and that there is a risk if you're lowering too much, you're going to now give somebody Alzheimer's. So you have to really be region-focused, not trivial, but we are pursuing such strategies where we found that because wild-type ataxin-1 is less important in the cerebellum, if you lower both enough in the cerebellum, Larissa showed that using a microRNA strategy, that's potentially an approach using gene therapy. So we're pursuing that now. But I just wanted you to go through this tour to see how challenging, and when you hear trials failed, you can see now all the reason why they might fail if we're hitting both alleles, like the ASO and Huntington, for example, or where it could be regional effect. Now I'm going to switch to share with you how my work on SCO1 really informed my thinking about Alzheimer's disease. We talked about the importance of protein levels, and we already know this from many human studies that the levels of tau, APP, and synuclein matter. The genetic studies tell us that. So I'm going to focus on tau today, and I'm more and more interested in that as far as perhaps controlling the levels of tau as a strategy for Alzheimer's disease. So I'm going to share with you this piece of data. It's unpublished, but it's a beautiful piece of data that tell me my dream for one day preventing Alzheimer is well placed. So this is looking at young mice and old mice, and looking at tau levels. Tau has multiple forms. Some forms, when they're on the microtubules, they're safe, they don't hurt the neurons. And some forms, when it comes off the microtubules, it gets phosphorylated. That's when it aggregates and starts wreaking havoc in the brain. And there are antibodies that detect these abnormal or toxic forms of tau. And I was curious, as the animals age, and would that really affect tau levels? And to my surprise, even in healthy mice, as they age, you can see they don't have these toxic abnormal conformation forms of tau. But you see it at 12 months of age. It actually starts at 6 to 9 and it gets worse. Whereas other forms of tau that are not toxic, they stay the same. Here's another form that's known to be quite toxic, and you'll see it accumulating as the animal age. So if this is happening in mice at 1 year of age, you can imagine what's happening in older humans. And I think there are data to suggest this is happening. So it's really this observation that sort of inspires me to find genes and pathways that regulate tau levels. And here, if we can find safe way to inhibit some of these pathways, and we can have a small molecule, a slightly lower tau by maybe 20%, we can perhaps, as we have polygenic scores to identify people's risk for Alzheimer, that would be an approach that one can take it when they're healthy, when they're 40 years of age, but have the risk. They can take this just like people with high cholesterol will take a statin when they're still healthy, not when they've got a heart attack. So this is my sort of dream for Alzheimer's disease. And we've done multiple reiteration of finding these regulators. I would mention to you one that was really exciting that was discovered by Christian Lasagna when he was a postdoc in our lab, where he found this kinase that enhanced the phosphorylation and get when tau is off the microtubule and causes degeneration, and inhibiting this kinase genetically improved the pathology in a tau model. And I love this piece of data because physiology doesn't lie. You can say behavior is soft, pathology is according to the eye of the beholder, but not physiology. This is in vivo LTP. And this is depressed LTP in vivo in the tau mice, but here they are when Christian lowered NOAC1 by 50%. And we are actually continuing to follow up on his work, reproduced it in our lab, and working with UCB Pharma to potentially, if we can identify a good inhibitor for this enzyme. But from that same work, we identified another kinase, and this is tyrosine kinase 2. And to our surprise, nobody has linked this kinase to tau. And this is now work done by Jiyeon Kim. It's all new and unpublished. We went into the brain and we reduced this kinase, and you'll see very nice reduction of tau levels in the brain. When you can see it also here. This is tau levels nicely reduced. So always when you're doing that, you want to understand how is this kinase affecting tau levels. There are only 5 tyrosines in tau, so that was really a blessing. And when we overexpress TYK2 and use a pan-tyrosine antibody, we see this very nice phosphotyrosine of tau. If we start mutating these phenylalanine one by one or mutate all together, you'll see if you mutate them all together, you express TYK2, you don't see any phosphorylation, but there was only one of them, 29, that when mutated, we lost the phosphorylation by TYK2. And I should mention, there have been other kinases that phosphorylate some of these other ones. The only tyrosine on tau that's never had a dedicated kinase was this one. And this came from unbiased genetic screens in cell and fruit flies. So with that, G then went on to see, does this kinase affect tau levels? And you see here we have a inducible system where you induce tau and you turn off the tetracycline, and the tau will degrade gradually. But if you overexpress TYK2, you prolong the half-life of tau, so you stabilize tau. However, if you mutate this tyrosine, TYK2 no longer has an effect. So it's clearly phosphorylating this tyrosine, and it's clearly affecting the stability of tau. And when we went then in vivo into a tauopathy model from Virginia Lee, you could see here the negative control tau levels, all forms, remember these toxic forms of tau, you'll see all of them have reduction when we lower TYK2. And then when we go in and look at the pathology, again, you could see gliosis is reduced and astrocytosis is reduced in these animals in all brain regions, cortex and other brain regions we looked at, but this is showing you for the hippocampus. So this is really quite exciting because here's another kinase came in an unbiased way that tells us this also regulates tau. We know that individuals with loss of function alleles who are healthy, but if you lose both alleles, there's some autoimmune disorders. So now we have two kinases. You can either maybe lower this one by 50% or very partially lower NOAC and this one together, combination therapy. These are potential strategies to reduce tau levels. So having done this on the kinome, and this is how we discovered these genes, we decided to go whole genome and find as many regulators because they'll teach us about tau biology, but they'll also maybe find us even safer and more interesting pathways. And with that, we identified several, and G then confirmed those on endogenous tau level, took 16 in vivo and showed of those 16, I believe she confirmed 13 of them lower tau in vivo. And she then went on, I'm just gonna skip here just to show you an example, how she did it in the adult after birth using tetracycline inducible. So when you induce tetracycline, you'll see here reduction of tau when you knock out this particular ubiquitin protease. And this is just to show you that in addition to lowered tau, we see correction of behavior. So this is a contextual fear assay where the animals, if they're shocked in the same cage twice, they'll freeze the second time. If you lower any of these candidates here that regulate tau, freezing is normal, but you can see the tau mice, this is healthy mice. The tau mice don't freeze as much because they don't learn, but if we lower tau levels, the animals freeze. And the same here, we see decreased gliosis in these animals, and we find reduction of all forms of tau and toxic forms of tau. So we focused, quite a few of them came from the ubiquitin pathway. This is a pathway that degrades tau. And we knew about one ubiquitin ligase that degrades tau. So this is involved, and this was discovered in Michael Hutton's lab, who's here, VP of Research at Lilly, and Leonard Petrocelli, who was in his lab. So CHIP is a ubiquitin ligase for tau. And what we discovered is we knock out these two other ubiquitin ligases, we see CHIP is increased. So these two ubiquitin ligases are degraders of the degrader. So this degrades tau. And now the reason we got benefit from inhibiting either of those, because we stabilize the degrader of tau. And I will just show you what we learned from all these studies. CHIP ubiquitinates tau to degrade it. The ubiquitin protease antagonizes this. Therefore, when we inhibit it, we preferably degrade tau. And then these ubiquitin ligases lead to the degradation of CHIP, which stabilizes tau. Therefore, inhibiting them is the reason we saw rescue, and this was an upstream activation of these ligases. So with these, now we have three beautiful candidates. Those two, we know that their loss of function is protective. I mean, not protective, but safe. They're patients who have individuals, healthy individuals that have loss of function allele. So we believe if we found an inhibitor for those, that will be safe. Also, activation of CHIP we're excited about because CHIP turns out to be a degrader of many misfolded proteins. Haploinsufficiency of CHIP causes one of the SCAs, SCA48. So if we can somehow stabilize CHIP, we will help not only Alzheimer and tau-induced pathology, but perhaps many other. So I'm putting all of my force in this direction now because of the broader implication of enhancing this pathway. In the last 2 minutes, I'm just going to share with you a really exciting story that's new, totally unexpected, that might help us understand regional vulnerability in tauopathies. And this is work by Chloe Chung. So Chloe is a postdoc in the lab, and we all know that tauopathies and dementias progress, affecting the cortex predominantly, and somehow the cerebellum and the brainstem are spared. So why are they spared? And so the beautiful blot about the aging mice I showed you was from Chloe. And when we were looking at that blot, we noticed there was one form of tau that did not accumulate with age. And that form of tau is right here in the cerebellum. It's a much larger form of tau. And if you look at all the splicing and all tau isoforms, everybody focus on all of these forms. But there is one that adds hundreds of amino acids to tau if it includes these two exons that somehow no one studies. It's been discovered probably 20 years ago in the dorsal root ganglia, studied in the rat DRGs, but no one pays attention to it. No one has studied it. And we notice it's more in the cerebellum and not in the cortex and hippocampus. And we wondered, it's a correlation. Could this be contributing? And we quantified it now in human patients. This is mice, this is human. We made an antibody to this long isoform, and you see it highly abundant in human tissue. And here the RNA-seq data, you see how it's much higher in the cerebellum compared to the cortex. We wonder, could this be really the reason you don't see pathology? So to do this, we are doing multiple studies, but I'll share one with you. If you take tau and add recombinant tau seeds to it, and then separate the proteins using detergent, the toxic form of taus will be detergent resistant. So if we do this, you'll notice here first in the soluble, you'll see big tau and regular tau 441. But if you add the seeds, you only see regular tau aggregating in the soluble, in the insoluble fraction, not big tau. And without seeds, of course, you don't see either, and you see the quantification. So having seen that, Chloe then went in vivo, and she expressed mutant tau, either big tau or regular tau, and looked for insoluble aggregated tau. And we see that for 441, but we don't see it with big tau. So we believe this big tau is probably playing a role, and we're focusing now on the mechanism. How is big tau resistant to aggregation, and how is it possibly protecting? One thing we've done, we knocked only big tau from the mouse brain, and now we can go back and see, do we make the cerebellum vulnerable? And do we accelerate the pathology in the hippocampus? So this is ongoing work, but this is, again, for the student, it was really purely from an observation on a Western that something is different in different tissue that Chloe predicted. So I will conclude with this slide. I hope I've shared with you data to convince you that digging deep in the pathogenesis is helpful. And here are some unexpected results. I did not expect to have the same complex cause degeneration by two different mechanisms. I thought it would be one mechanism throughout. I was humbled by that. And the same mutation in widely expressed protein, even for the gain of function, it's not only one mechanism. So forget the two regions, even within one brain regions, different cells, probably different interactions. And I hope this is an exciting result I share with you. I hope you're convinced protein levels matter. And the cross-species studies for us were really helpful. When you go across billions of years, the biology is conserved. And with that, I'd like to thank the families who participated in our studies, our funders, particularly the JPB Foundation, who really believed in our work and allowed us to take risks. And I mentioned all the people who's contributed to this work. I'll just show you the faces of all who've contributed to the work. Never one picture captures everyone in the lab. They're constantly graduating, which is a good thing. And really, this award is acknowledgement of all they contribute. But G did all the recent regulators helped by Bajos, who's the one I'm convincing to apply here. And this is Chloe, who did the tau studies, and the others are the SA1 team. Thank you very much. Thank you.
Thank you so much for absolutely a beautiful, beautiful presentation. We have 2 microphones here, and anyone that is listening can, on Zoom, can actually put things in the chat, and we have someone monitoring that. I kind of want to start off with 2 questions. So the first is I wanted to have you highlight once again how important finding negative results or unexpected results turns your science around. And I think this is a great lesson for all the trainees who get disappointed when that blot doesn't work out, but sometimes it's a blessing.
I totally agree with you. As you could see, some of these negative results, actually our negative result with SCA1, my student was so depressed when she found the knocking didn't rescue. It became the topic of an R01 that gave me a 1 percentile, because they said, you know something, no one had looked at regional vulnerability in an authentic model. So that negative result is leading to a future research direction. Just tangible example.
Yes, yeah, an important tangible example. Since this is a CTSI, I was hoping that you might be able to take your initial findings of your ataxia and actually bring it back to the patient. So these are patients who are normal at birth and then develop the disease, yet it's a genetic defect. And I think we often say, oh, it's genetic, it has to be there at birth. And this is a good example.
I think, yes, this is very important. They live healthy for about 40 to 45 years, unless they have very big repeat, then it shows earlier. And that's, to me, an opportunity for therapeutics. But I want to take it back to the patient in another way. I had a family, I didn't have time to show you, this family that led me to map the SCA1 gene and clone it had 200 affected individuals in it. It's from Montgomery, Texas. I went and examined all of them to get the blood. Many, because you have kids before you know you're affected, so you have so many. Today, I don't think I can have a person affected in this family. So when we do a clinical trial, we can't treat people in this family because we eliminated the disease. It was another situation for a patient of mine in Chile. He had pre-implementation genetic diagnosis and was able to have 3 healthy children and 2 healthy, twins. And basically the disease ended with him. It was multiple generations in Spain in his family. So even though we haven't discovered a really effective treatment that reverses the disease, genetic counseling work. If you let these individuals know they have the disease, they can plan, they can have, pre-implantation genetic diagnosis, they can have sperm donors. There are many options today. So that's how we eliminated the disease in the Houston ataxia family.
Howard?
Lovely work. In terms of the big tau versus tau, are you getting any hints from AlphaFold in terms of what these proteins look like?
Great questions, very appropriate, since the LASCAR was just given to the scientists who developed AlphaFold, as we heard from the recent announcement. We've tried, and we're getting a little bit of hint in that we're finding some low complexity sequences in that. We're testing those specifically to see how this affects the accumulation and aggregation in specific assays of the disease. So that's one thing we are doing. Good question, thank you.
No. So another question for you is back to your Rett's syndrome. So where are you with those discoveries?
Thank you. So with Rett, again, the early diagnosis, now the diagnosis has shifted. It used to be Rett doesn't develop right away. You lose one symptom at 12 months, another function at 14 months, and so on. So you had to wait 7 years till the full syndrome is established to give it the diagnosis of Rett. That was the clinical criteria. Now we accelerated that. Most cases are diagnosed at 3 years of age, so they get better therapy. But we learned 3 things that are really important. We learned the dosage of the MACP2 protein is really critical. That's a Goldilocks protein. If you change it by 30% in either direction, you got neurological symptoms. And with that, we discovered first in mice, and then it was validated a year later in humans that doubling the level of the protein causes a devastating neurological disorder called the MACP2 duplication syndrome. Sadly, these boys die by usually third decade or so. So they have terrible seizures, very severe progressive disorder. We did 10 years of work using preclinical proof of concept studies in humanized mice using ASO. We found you can reverse the disease in adulthood, even in mice as old as 9 months of age. And I'm very thankful for Ionis Pharma for now moving those studies, and hopefully clinical trials will start in Q1 2024. They're doing the natural history study, the clinical trial readiness study. We're hoping 2024 the trial will be. So that's one. We learned something in rats. Remember this period of normal development? We discovered mice have a period of normal also. We discovered that if you actually train the animals excessively as in like every 2 weeks on the same task, you can actually get them to perform normally as they age, and you can delay the onset of disease by a month. And this would call for newborn screening, because you have 3 years where you can train these patients. The rat neurons are there. They function normally, no degeneration. They just function at reduced capacity. Physiologically, rather than 100%, they work at 70%. It turns out this extra training increased their plasticity, increase their dendrites, correct many of their physiology and features. So we're hoping some newborn screening will be online soon. And even if this is not a cure, when better therapies, gene therapy has to be carefully managed because you have half of the cells with normal protein. So you don't create the duplication in half the cells. Hopefully when we have great therapies, even if we diagnose early, they probably will respond better to the therapy.
I was so struck yesterday when you were speaking with our case scholars about your institute and the importance of the interdisciplinary work. And I wondered if you could comment about that, not only the team science aspect, but sort of how you found that team science really works and can be fostered.
Sure, thank you so much. So, after I've done some progress with my work with RET and SCE1, I was approached by Dr. Feigen, then chair of pediatrics and Texas Children's Hospital board member. They said, you're successful for what you've done for RET, but how can we do this for a lot more neurologic diseases? And I reflected what made me successful. What made me successful is the interdisciplinary approach to the diseases. I don't stick with the boundaries of what I'm familiar with. I find scientists around me that I recruit to help me, whether it's flies, it's in vivo physiology, things I don't know how to do, but we all work together. And that's the plus from living in a generous environment, collaborative, which you all have here. So with that, I thought, okay, well, that's what works for me. If we did that for all neurological diseases, think about the impact. And how do you do that? I thought if we build an institute, it doesn't always work. Not everybody is as outgoing as I am, who's going to reach for anyone, no matter where they are. For some scientists, you have to bring it to them. So I thought if we build an institute and hire people from various disciplines, provide core facilities. So if you're shy and you don't want to go work with another investigator, you just want to use the core facility to do behavior, physiology, etc., that would help. So we did that, and it opened about 12 years ago, and recruited 25 assistant professors. We made sure they had that ingredients of generosity, collaborative spirit. They were recruited over 10 years. And basically magic happened. Over 45 unsolvable diseases were solved. They were solved because collaboration between a clinician and a Drosophila geneticist who's engineering the unknown variants of unknown significance into the fruit fly to see if it's pathogenic or not, defining pathways, going back to eye neurons, going back to mice, and so on and so forth. To our surprise, clinical trials ensued from these studies, and we've got some that were completed, everolimus in catastrophic epilepsy. We've got one ongoing, ASO for Angelman syndrome. You heard about the ASO coming online soon, and so on and so forth. So that's really what happens when people from different disciplines work together. We have 9 different departments among these 33 faculty.
Well, thank you for your inspirational talk and your inspirational work for on behalf of all the families that are suffering from these horrible diseases. So let's give another round of applause. Thank you. Brilliant.